Welcome back to Neural Frontiers. Today we are diving into one of the most challenging aspects of neuroscience drug discovery, validating targets in the brain's incomprehensibly complex network. Traditional drug discovery relies on one target, one disease paradigm. Yet the brain operates through interconnected networks where single nodes can influence multiple circuits. How do we validate targets in a system where perturbation of any component can have cascading effects throughout the entire network? Are we approaching neural drug discovery with fundamentally flawed assumptions?
That's a great point. Unlike organs with simpler pathways, the brain operates more like a vast orchestra. Changing one instrument often changes the whole melody. Traditional one target, one disease, thinking that doesn't capture the complexity. We need to embrace system level approaches, use network biology, and consider not just how a target functions in isolation, but in the full context of neural circuitry. Computational modeling and advanced imaging let us watch these cascade effects in real time, helping us validate targets within living neural networks, not just in petri dishes.
Right. So the reproducibility crisis in research has been particularly acute in target validation studies. Mouse models often fail to translate to humans, and cellular models often lack the complexity of intact neural circuits. Given these limitations, how do we build confidence in target selection? Should we demand higher standards of evidence before advancing to clinical trials?
Absolutely. The reproducibility crisis is very real in neuroscience. What works in a mouse model often fails in people, and sometimes our cell cultures miss crucial connections present in the actual brain. So, to build confidence, we need multi level evidence like genetics, cellular, and physiological validation, plus confirmation in models that capture the human brain traits. Higher standards like requiring evidence from multiple models and using tools like human iPSC driven neurons or organoids are essential before advanced clinical trials. It's not about just checking boxes, it's about really knowing our target matters in disease.
Right. So let's talk about polypharmacology. So, it is indeed that drugs acting on multiple targets has shown a lot of promise in neuroscience. Yet, regulatory framework favors single target specificity. So, how do we develop and validate drugs which are designed for network effects rather than molecular precision? Are we handicapping ourselves with reductionist approaches to complex systems?
Neuroscience is showing that drugs acting on multiple targets, which is polypharmacology, may be key to treating disorders involving entire networks. But regulatory frameworks are still mostly tailored to single targeted drugs, partly because they are easier to characterize for safety and efficacy. For true new network targeting drugs, we need more validation strategies, mapping out network effects, building computational models, and collecting more dynamic biomarker data. The hope is that regulators will evolve alongside the science, allowing us to harness these approaches while still protecting patient safety.
So, the human brain's unique features, such as enlarged prefrontal cortex, extended development, complex language circuits, have no true animal model equivalents actually. So, how do we validate targets for uniquely human neurological and psychiatric conditions? Are we fundamentally limited to our ability to model human brain disorders?
You're right. The human brain features doesn't exist in any animal, and that's a huge challenge. Conditions like schizophrenia involve circuits that only humans have, That means we are fundamentally limited. The best we can do is use human based models like stem cell derived neurons, brain organoids, and integrate human genetic data. These models aren't perfect yet, but they help bridge the gap. Collaboration with imaging, genomics, and even postmortem tissue studies is helping us edge closer to understand these unique human disorders.
So, biomarker development in neuroscience lags far behind oncology, other therapeutic areas. Without reliable target engagement markers, how do we know if our drugs are hitting their intended targets in living brain? What would it take to develop a robust biomarker ecosystem for neuroscience drug discovery?
It's a big problem. Without reliable biomarkers, we can't be sure our drugs are hitting their targets in the brain. Neuroscience lags behind these fields like oncology here, partly due to the brain's complexity and inaccessibility. To develop a robust biomarker ecosystem, we need advanced neuroimaging, better fluid based markers from blood or cerebrospinal fluid, and sophisticated digital biomarkers from wearable devices. It will take collaboration between academia, industry, and regulators to standardize and validate these tools, but it's essential for future drug discovery.
Thank you, Jhansi, for joining us in our podcast, Neural Frontiers. And thank you to our listeners for listening to our podcast. If you like the podcast, do not forget to like, share, and subscribe.